How can you know? I mean, no, not many people. I am online. Hello. Yeah, thank you. Thank you. Okay, the attendance is not at all there. Anyway, so let me start. Good day to all of you. And I can see Krish. I already had wished him, and he also wished me. Anyway, so hope you are keeping fine, Krish.
Thank you.
Right. Yeah. So this session gets recorded. So those who are probably busy with their professional schedules may not be able to join us at this moment. I understand. But okay, the session will be recorded. So let me just go through some of the things that we discussed in the last few sessions and modules, divided into various modules. So let me highlight about module number 6, where we had discussed about the industrial and pharmaceutical applications of microorganisms. We started with an basic lecture on microorganisms in industry. We try to discuss about the various aspects of applied microbiology, particularly with respect to industrial production overall. Then we went on to discuss the application of microorganisms in the pharmaceutical industry, because that is one of the most important sector, and also concerned with human health. And then we also discussed about the molecular enzymatic applications of in industry. So enzymes as single molecules are also very, very important, apart from microbes as a whole, or microorganism from the industrial point of view. And then we discussed about engineering microbial consortia for industrial applications. So mostly, many of the industrial processes are driven by single species. For example, Saccharomyces cerevisiae is important in the fermentation industry, both in the alcohol production as well as in the bakery industry, for example. Then we have lactobacilli, which are important in the milk industry. And there are several others as microbes, which are used as pure, species, or particular strains are used for carrying out certain industrial applications. But then there are certain industrial processes where a mixture of microorganisms are used to achieve a output. We start with a set of substrate, and then we use a mix of microbes, and then these microbes may work in association with one another, or they may work in a sequential manner, and then they give us a final product. So many of these things have been done with natural population of microbes, and today we try to increase the efficiency of such processes by engineering such natural populations. So that was one of the important points we discussed under this industrial and pharmaceutical applications of microorganisms under module 6. Then we had module 7, where we laid emphasis on the application of microbes in the agriculture sector, particularly in the production of biofertilizer, because nutrients are very, very important for plant growth and development, as well as giving us quality agro products. Then we also discussed about microbial pesticides, because we try to get rid of chemical pesticides which are very, very harmful to humans and other higher mammals, as well as the environment in total. So microbial pesticides have turned out to be a very important input in the agricultural sector, where we can control pests effectively with biopesticides or microbial pesticides in particular. And we also discuss about the integrated pest management approach, because a single method may not be effective in controlling the pests 100% overall. But if we deploy a combination of various methods, particularly the physical methods of control, combine it partly chemical and preferentially, using microbial pesticides, and also, what we call as biological control, can actually give us very, very high control of microbial, pests and diseases in agricultural plants. So this was the focus of our module number 7. Then we had another module following this, which is basically on environmental biotechnology. Here we discussed about the microbial ecology and the various biogeochemical cycles and their importance in maintaining the ecosystem overall. Then we went on to discuss about the application of microbes in treating waste, particularly city waste, urban waste, as well as also agro wastes. So there are physical methods, for example, like burning and incineration, which can take control of wastes. But those are highly energy intensive, and they also cause pollution. But nevertheless, they are used under controlled systems and have been very, very effective. But then there are also chemical methods whereby you use a lot of chemicals to degrade certain waste. That doesn't provide us solutions for the wastes. So one of the important area is using microbes in the treatment of waste, whereby the microbes use the waste for their own metabolic activities and derive energy and matter from the microbes, transforming them into harmless chemicals, and also sometimes producing value-added products. So from that point of view, microbial waste treatment methods was dealt with extensively, and this can help us in converting waste into wealth at a very low cost, because these are not energy intensive, but of course we may require to build up certain infrastructure for carrying out such waste treatment methods using microbes. Then we also discuss about microbial bioremediation. So we do microbial waste treatment in a confined environment, but certain times it happens that there is a hazard which occurs due to oil spills or some kind of spillage of hazardous chemicals while being transported, and it's out there in the wild open. So those are dangerous for human as well as animal health, as well as they also damages the overall ecosystem. So how to treat such spillage sites? So for that, microbial bioremediation is one of the most important approaches. Then we had the module number 9, where we laid emphasis on the food production using microorganisms and also the various high-value products and health beneficial products that are derived from microbial fermentation of raw agro products or any other food products. So we discussed the founding principles of food fermentation, and we also discussed about the various health benefits as well as the risks involved, because sometimes these processes, if they are not efficient, may actually end up with a microbial load and may harm the consumer. So we need to know about those instances as well. So apart from food, beverages are also an important sector when it comes to application of microbes. And today now various biotechnological innovations has been made for making these processes highly efficient. And then we also covered one important topic, which is the single cell protein production. And these are having high commercial value, as well as many of these methods actually operate by the principle of converting waste into wealth. Then in module 10, we discussed about the application of microbes in medical biotechnology. Although we have dealt with the application of microbes in pharmaceutical industry, here we laid emphasis on production of biopharmaceuticals like enzymes, antibodies, and therapeutic proteins using the recombinant DNA technology. And we had an extensive discussion on the production of vaccines. Then another important area where microbes are playing a critical role is in the generation of alternative energy. Due to the climate change challenges emanating from usage of fossil fuel, which contributes to the build-up of greenhouse gases. Today, microbes are being looked as a promising tool for producing biofuels. And then in a more advanced applications, microbes are being used in fuel cells and also in the production of microbial electrolysis cells. So in this module number 11, we discussed these topics in depth. Then finally, in the final module number 12, we laid emphasis on patenting in microbial biotechnology. In all the other 11 modules, starting from the foundational aspects of microbiology and biotechnology, and a mixer of the two disciplines to produce, desired products, whether it is in the industry or whether it is in the agricultural sector or it is in the medical sector or in the production of food and beverages. There are innovations made by various scientists through the ages time in recent years, and because they hold commercial potential, it is very, very important to protect them from the point of view of intellectual property rights. So these module, we started with an introduction to intellectual property, then patent laws, biotechnology patents and the application processes, and of course the legal and ethical considerations. Patent laws are actually bounded by the national geographical boundaries of each country. So they differ from country to country. But overall, they have certain commonalities based on which these laws are enacted by each and every legislature or the parliament of every country. So we try to discuss about the common things in the IPR field, particularly with respect to applications in the microbial biotechnology field. So with this, we have covered the entire modules of this course. Now, we also had the live sessions, which was attended by a few of you. And today also we are having this live session once again. However, unlike last time where we had many queries, this time we are not having much queries. I'm not very sure whether it is due to the busy schedule of the students or learners, or it may be some kind of other symptom. Anyway, we got two questions. Let me get into those questions one by one. So once we end up discussing these questions, I will try to have some discussion with the people present over here. So I'm not very sure whether the people who put these questions are there in this group. But anyway, let me try to go to the first question, which was sent by Prabhu.aut.26@gmail.com. So his question is about biostimulation. He's asking, what does biostimulation approach in bioremediation primarily involve? This is already discussed over there in the lecture. So let me once again repeat. So there are two words which has been combined to produce this term. One is the biology, and another is the stimulation. So together coming as biostimulation. So basically here we are trying to stimulate certain biological entities by some means or interventions. Okay. Now where we are using these biostimulation approach in the process of bioremediation. So bioremediation is about in situ correction approaches. So where we try to rectify the problem that is happening on site. So even if we do not do anything and leave that place for long time, the natural microbial population will take care of that site and will degrade it. And then, of course, other natural processes like microbial transport due to natural processes may happen, and that site itself can act as that contaminants in the site can act as selection pressure to establish certain strains of microbes over there, which will slowly degrade these contaminants on the site. But we cannot wait for such long time for the contaminants to degrade, because we need to, in certain cases, we may need to utilize that land or that site at an early date for various uses, for building up residential complexes or any public utility. So we need to remediate that site, and also that those contaminants may actually pollute the water bodies in the ground below. So we need to actually clear them and make the groundwater safer. So for doing this, what we do, we try to increase the activity of the indigenous microorganisms which are present at the contaminated site. So this can be done by supplying them with nutrient oxygen and other conditions that promote the growth of these indigenous microorganisms. So if you tell what is the approach, primary approach, primary things that in bioremediation, when it comes to biostimulation, we basically facilitate the growth of the indigenous microorganisms by supplying them with nutrients, okay, which may be certain minerals or some kind of cocktails having enriched with nutrients, And then we also supply oxygen to promote their growth, and also any other conditions that will promote their growth. For example, certain we know certain organisms grow better at higher pH or lower pH. So we may be manipulating those pH conditions. So this is what is the primary approach in biostimulation. So anyway, let me go to the next question, which is from Raju PSPK 1245 @gmail.com. So he has a very straightforward question. He's asking, what is mitigation? Okay, before going on to explaining what is mitigation, let me tell you something similar. I mean, not similar exactly, another approach, which will help us in understanding the exact approach of mitigation. So, for example, when there is a degraded environment, there is a shortage of nutrients, and we may see that at a macro level. For example, the forest cover is disappearing, the grassland is also getting reduced. So for survival, the animals have to adapt to that and changing situation. So that's called adaptation. Or there may be changed vegetation in this system, and those vegetation are actually not favored by the animals residing in that locality. But then the animals have no choice but to start eating those vegetation which has started dominating that area. So that's what we call as adaptation. So similarly, that's what is visible to us, but in the unseen world, the world of microbes, we can similarly see, for example, whenever there is a spillage, for example, in the earlier discussions when we were discussing about the bioremediation, so there is a concentration of a lot of contaminants in a site, which may kill many of the microorganisms on site. But some of the microorganisms may be able to adapt to the changed situation and somehow survive. And we have discussed about many of these mechanisms. For example, sporulation can be. So this is what we call as adaptation. Adaptation is a process where the microorganisms or the macroorganisms try to adapt to the changed environment on their own. Okay, but mitigation is something different from here. This adaptation. In mitigation, there is a human intervention. Okay, where we try to manipulate the environment itself, improve the environment to make it more habitable. For example, in the case of bioremediation by biostimulation, it can be considered as a mitigation step. Okay, so to explain it properly or in a formal way context, we can tell that mitigation refers to action taken to reduce or prevent adverse environmental impacts, particularly by reducing the intensity of harmful activities or by enhancing the environment's ability to absorb those impacts. So in microbial biotechnology, mitigation is done by using microorganisms or their byproducts to reduce or eliminate harmful effects of pollutants or environmental issues. So I hope this makes the term mitigation clear, because when we compare it with adaptation, we can understand these are two different things. So with this, we come to end of the questions. which has been mailed to us, and this has been mailed back to the students, and also we have put up for discussion here. Now let me try to have a discussion with the people available over here. So now I open this room for a discussion. Yeah, if somebody has some query, you can raise your hand and ask me the question, or you can also type. Yeah, Chris, please go ahead.
Hello. I wrote into the Google link that we got the email from, and I put my question into that for this, the few questions I had. I'll just go back and have a look at a few. I can read them out to you. One was on sonication. One was on sonication, on the frequencies and the, what's the other particulars I wanted about that one? The sonication frequencies. Yeah, that's what I've got written here, but just some information about that related to bacteria and microbes too, because it was brought up in one of the lectures, and that really interested me. And I thought, well, people use bug zappers, and with their electronic bug zappers they got, this might be interfering with our organic microbes that we try and produce. So it's something that I want to learn more about. Over.
Yeah, can you tell which lecture you are referring to? Lecture number, module and lecture number?
No, not at all.
Yeah.
It was written down a good 4 weeks ago. So I've been meaning to ask this for a while. I've got it on my calendar here. I got a sonication. It was briefly mentioned in the end of the microbe lectures. So that would have been week, here, let me have a look. The agricultural week, which was week 7. I presume it would have been in that week. Module 7.
Okay, Chris, is it in module 6, lecture 1, where we are discussing about downstream processing under cell disruption?
Yeah, yes, that would be, yeah.
Ultrasonic disruption?
Yes, that
okay,
sounds like
so,
it.
Yeah, that's fine. Yeah, so exactly what's your question regarding here?
You must have some studies or something you've got this information from. So I just want to know that the frequencies that interferes with the microbes, like they might say 1,000 or 100 kilohertz might interfere with the processes of the pathways of certain microbes. And I'm just interested in those figures, really, those numbers, to know with the ultrasonic bug zappers that people use, they emit certain frequencies to kill insects off, and that might be hurting our microbes, which are very important. So I'm curious on just the frequencies itself. Like they might say Bacillus is known for 25,000 hertz frequency that it lyses at, for instance.
Oh yeah, I think actually you're telling about ultra high frequency?
You're correct, yeah.
Yeah. Now here the process actually need to be organized based on the mass that you are having. The process, the principle is very simple. We use optimize the frequencies which can disrupt the cell, and once the cell is open, Basically, our aim is to open up the cell, which can be through lysis or which can be kind of mechanical disruption also, and biochemical lysis as well, or chemical lysis, or due to osmotic pressure. So if we increase the target pressure inside the cell, it will burst out. So another physical method is using this sound waves. So they will disrupt the cell membranes, and then as a result of which the products which is inside the cell will be released into the buffer, and then we follow up other cleaning steps by which we do the product recovery. So yeah, anything more specific with regards to this?
Yeah, the frequency, the actual number that's dialed in to get the cell to lyse.
20 to 30 hertz can be optimum for many of these processes, but then actually the bacterial cell and the yeast cell, they will have different kind of optimization processes. So it will actually depend on the microbe that we are using for producing the metabolite. So that's actually needs to be optimized for each and every type of microbe.
So so 330 hertz. So it's really low. Like that's approximately the frequency they use nearly in the base spectrum.
No. There are several things here actually. So for example, we use a certain buffer. Okay. So it's a combination of many of these things, the target pressure as well as the frequency employed. So it's not just, but of course, in certain cases we are only using the lysis buffer. We are not using sound, right? So now this is additional, which helps in that process.
Okay, it's combination of the frequency with everything else to get the outcome.
Yes.
That's a shame. I was really hoping you were going to say yes. With 25 kilohertz, it destroys this, this genus, and I've been, wow, this is really cool. Then I can add that into my studies and that sort of thing. Thank you for clarifying that. My second question was on a buscular mycorrhizae. What's some types that I could source, that I could use in my organic garden? What's some species or genus, I suppose, that I could look for a good buscular mycorrhizae? Because I got various other types that I use, and I particularly source the ones with phytohormones as well. So they'll do two tasks for me, and I just thought I'd ask that question about the buscular mycorrhizae. Over.
Yeah, can you refer to the module that will help me actually tracking out the content, and then we can discuss.
Yeah. All right, I'll have a quick look for you.
Or the lecture title, if you. Microsin, yes. Which part are you asking? Microsis biofertilizer.
This is the one basically the microorganizer.
So are you referring to module 7, lecture number 1? It's a question is on biofertilizer, right?
Yep, there it is. I see it too. It's slide number 43.
Yeah, arbuscular mycorrhizal fungi.
Yeah.
Right, exactly. Oh, fine, that's fine. What's your question actually?
What's a genus or species that I could source to use in my collection?
What genus or?
species. What's it like? What's a microbe? What's a species of fungi of buscular mycorrhizae that I could source?
So that they can use and source in his garden.
Because I've got a fantastic list that I use at the moment, but they just work amazing, and getting them to work for you and do specific tasks, I'm having great luck.
So you were looking for certain species in particular which can be used for this process.
Yeah,
as I can understand.
Yeah, that's correct. Yeah.
Okay, let me try to get a list for you. Yeah.
So given the complexity of the questions, I think we can look into it and answer it.
No.
Just its location, all these things.
Yeah. So actually, so some of the dominating species, it actually depend on the location also. So you may have glomus species particularly. Okay.
Yeah.
So,
I use
glomus. Am I audible? GLOMUS, glomus.
Yeah. Rhizophagus. Yeah.
Glomus species, but actually,
Rhizophagus irregularis. That's the that name got changed to glomus. I used that one.
You are using glomus.
Correct.
But in your location, what is the species found would be actually, that would be very, very location specific. Okay. So some of the common species that are reported are glomus cynosum. Okay. Then you may have macrocarpum. Then you have glomus clarum. So many. So some of the approach is to actually, enrich, go for diversity study in your ecosystem. So your temperate biodiversity and a tropical biodiversity spot will have a different kind of species as well as some of the genus. So that's actually basically location specific. But then for local applications, you need to isolate it from your locality and then go for the enrichment. So that's basically the approach.
I'm looking for just specific. I actually get my microbes from India. I'm in Australia, and I currently get them from India. So Australia is very poor in their availability to microbes. So I'm one of the rare persons in Australia that has a very good selection of fantastic bioremediation type microbes, biopesticides, biofungicide, everything. It's brilliant, and I just need extra ones that I can source from my suppliers in India, where you are.
Oh, I get your point now. I get it. So no, it's something different. I have to tell you. Yeah. I mean, those are possible, but then there are. I think in some of the lectures I spoke about the microbial repositories. I'm not very sure. So anyway, so in India there are now 3 microbial repositories. Okay, as per international convention, the oldest one is MTCC, microbial type. You can write down. Yeah. So oldest one is microbial type culture collection in IMTEC, Institute of Microbial Technology, Chandigarh. Okay. Then there is another repository, which is a repository of agriculturally important microorganisms in Mao, which is an ICAR institute. Okay. Then there is another repository in microbial repository in Pune, Pune, which is in the institute called NCCS, National Center for Cell Sciences. So these three repositories, they have collections of many of these various kind of microbes. But then exchanging biological samples is actually regulated by our national law. We have a national biodiversity law, which isn't as per international convention. So you have to go through that regulatory process. But then actually, I will come to the other part. I mean, see, we actually know maybe 0.00 and several zeros one, right? So your statement that Australia is not rich may not be true, because how much of that has been explored, we don't know. Yours is a very unique landlocked, sea-locked island. It has a different kind of evolutionary history, and nothing much has been explored, I believe. What's the extent of financing or funding for really carrying out this expert work? So you may actually end up getting very promising species in your own land, because taking species from one place to a unfamiliar terrain, many of these experiments have actually failed to a large extent. So those are issues associated with this. Yeah, but anyway, collaboration, and then there are many regulatory frameworks which basically regulate but doesn't prevent. These things are possible, but it has to be done through the process. Yeah,
I've contacted our government in Australia. I've gone through the right regulatory permits that are needed for the imports, and I've done that. So I've got them in place. So I just need the right microbes now, the other microbes, because I've got I've got about a dozen that I use that I've proved to be amazing. And of course, the difference, you get different strains of the species, but my ones still do the job that they were supposed to do. So that's all I can ask. And most of these you can just get out of milk. If you isolate them out of milk, you can get them. But I'm not up to that stage of of isolation and repeating and that sort of thing. I'm in more of the plant science, not in the microbiology of the multiplying. So yeah, that's what I'm at. So it's so exciting and new, and it's great because the experiments work, mate. Microbes rule. Over.
Oh, that's excellent. Yeah. No, I mean, see, many we also get from the American type culture collection, so things are possible. I mean, it's just a regulation in place, because the other way around, many countries also have quarantine laws. I mean, your Australia quarantine laws are quite strict, as I know.
Oh,
so yeah. in your airport, you have to take out your shoes, right? So even dust particles not allowed.
Yes. They spray the airplane when you sit in it, when you land. It's all fumy and
Yes, right.
Weird
anyway, these are interesting discussions, and then a lot of commercial potential are there. So that's what this course is all about, trying to uncover the importance, commercial aspects of microbes, and so many different areas, including agriculture, health sector, industry. Yeah, thank you. Any other queries?
No, thanks. I'll reply to your email and talk further with you through the email. Because yeah, I appreciate your time as well. It's very valuable. It's helping me continue in my progress in my studies and my area of interest. Thank you.
Yeah, fantastic. No, so you are currently pursuing your doctoral studies or master's studies?
No, I'm doing it for personal interest. That's really, it's not for educational gain. It's for my own personal interest, because it's soil science plant science and microbiology, those three things are brought my area of interest, agricultural engineering, and I continued to do that through N. Patel, and I love it. And I put all those information, education to experiment out in the field out here on my farm, and I get amazing results, like massive fruit. I get annuals that perform, that keep growing as perennials. I get these weird things that's happening in plant science. It's fun and really interesting. Yeah.
Okay. Yeah. Thank you. All the best. Yeah.
Thank you.
So yeah, any other person in this group who have some other queries? If there are no any queries, so CET team, you may wind up.
There's no questions in the chat box.
Let me check if the chat box has some questions.
Professor, sir, we have answered those.
Have 2 questions? It's done.
No, just answer. So the questions that Chris asked right now.
Where it is?
So these are the questions that we just asked.
Okay. Yeah. All right.
That's it, sir.
I need to check in YouTube also. Any questions?
So there are no questions.
Okay. Yeah, I opened the chat box. There are no any questions over there. Okay. Then thank you, Chris, and all others present over here. So
could I talk to you off air after we finish, please?
Yeah. So if required, we can actually have one more meeting before the exams. So thank you, all of you.
Oh. Excuse me, N. Patel, I presume the professor has gone. Would that be correct? I was going to ask him another question off air. Oh, okay. Oh,